Welcome back, everyone. Our next topic is Warfen making a meaningful impact on the transfusion medicine ecosystem. For chairing this session, I would like to invite Dr. Mohandas Murugesan, additional professor in the Department of Transfusion Medicine, Malabar Cancer Center, Talasheri. He pursued MD in Transfusion Medicine from JIPMER in the year 2012. Sir has worked in Kannur as a district nodal officer in blood transfusion services. He has bagged Young Scientist Award at ISTM Tmascon in the year 2020. He is also a member of ISTM, ISBTI, ISBT. He has numerous publications in national and international journals. His main area of interest are apheresis, flow cytometry, research in transfusion medicine, and graft engineering. We welcome you, sir.
Thank you for the kind introduction. On the outset, I would like to thank the Transfusion Medicine Academic Society for inviting me for the chairperson for the session. So the topic, the next speaker will be Mr. Ajit Pal Singh. So he is representing Warfen India and is working as a regional sales manager looking after South and Western India. So he was also previously under Johnson & Johnson Private Limited, looking after the northern parts of the country. So he has an advanced program in sales management from IIM Kolkata. So he has an MBA as well as he has other specialities. So he was an organizing secretary of annual college fest for the past 2 consecutive years. He has participated in organized blood donation camps and for care for senior citizen campaigns with Rotary Club of Jamshedpur. So we welcome you.
Thank you, sir, for the introduction. And my name is Ajit, and I thank the organizing committee of TMAS for giving us an opportunity to come here and present the impact which Warfen is making in the transfusion medicine ecosystem. So before I begin, we would like to introduce our company with a small video.
We were a family-owned company founded in 1966 in Spain that innovates diagnostic systems, reagent, and data management solutions to enhance patient care. We are a worldwide leader in specialized diagnostics. With our headquarters in Barcelona, Spain, we operate directly in more than 30 countries and in more than 100 territories through distributors. With the acquisition of Immucor, we have 7 main technology centers around the world and 6 business lines: hemostasis, acute care diagnostics, autoimmunity, original equipment manufacturing, and now transfusion and transplant. Our purpose is to contribute to the advancement of patient care around the world through innovative specialized diagnostics. Our values have been guiding us for more than half a century. They shape who we are, how we work together, and define the behaviors that drive our success. Meet our president, Mark Ruberalda, and our CEO, Carlos Pascual. At Warfen, every employee can have a positive impact. We are one team developing diagnostic solutions that inform and drive critical clinical decisions. One team of 7,000 people around the world, powering patient care.
So there's just one announcement from the hotel side. One car, Nexon, is blocking the entrance to the hotel. KL07 CU 5977. If anybody has parked it, kindly move it from there. Thank you. Yeah.
So that was a small introduction of Warfen as a company. I will try to give you an overview of the products which we have. Some are directly concerned with transfusion medicine and some are indirectly concerned with transfusion medicine. So over the next 10-15 minutes, I will give you an overview. So Warfen is a specialized diagnostic company and we are committed to provide innovative diagnostic solutions. And we are leading player in platforms like acute care, lab diagnostic, autoimmune, and now transplant and transfusion. So we take pride in telling that every day, all the hospitals throughout the world are testing patient samples on our instruments, and this value is 1.8 million. So we take pride, and every day we are working, innovating to improve this number. So I'll just touch upon few of the solutions which we have in acute care diagnostics. We have blood gas analyzers. We call it a GEM series of products. We have advanced it. We used to have GEM 3500. Now we have the advanced GEM 5000. In the whole blood hemostasis segment, we have viscoelastrometric testing. The product is Rotem. And we also have a product called GemStat, which is a basic metabolic panel, but currently we are not marketing in India. We plan to bring it very soon to India. So this is about whole blood hemostasis. The products here is Rotem Delta, and the fully automated Rotem Sigma is also there. So we try to, the test which we do in this Rotem is Intem and Heptem in the intrinsic pathway, and Extem, 15M, Aptem in the extrinsic pathway. So basically, we as per the WHO, we are trying to make meaningful impact in patient blood management through this Rotem. And this Rotem, with this Rotem, it has demonstrated that in it has given us an idea of a very correct assessment of coagulopathies, and has advised us to go for meaningful transfusion or reoperations, and also help the patients and clinicians reduce the length of stay for the patients. So there has been a lot of studies and lot of experiences which have concluded that with the usage of Rotem, there is potential reduction in in-hospital mortality to the tune of 28%. The infection rate has been seen to have reduced by 21%. As far as acute myocardial infarction is concerned, there is a reduction seen to the tune of 31%, and also renal failure cases have reduced by 30%. So Rotem has helped us to reduce complications during and post interventions, whatever the clinician has decided. And also Rotem has been very instrumental in giving very guided treatment as far as transfusions are concerned. It has helped to minimize the inappropriate transfusion, like our earlier speakers were saying that cost is a very integral thing in the Indian health ecosystem. So Rotem has helped us minimize cost, minimize inappropriate transfusion to the tune of 41%. Also, we have a product called Verify Now, which helps us in whole blood hemostasis. The test which we are able to do with Verify Now is Prove test. It helps us to see the effect of P2, Y12 inhibitors, and also the aspirin test. And in the clinical setting, it has seen that Verify Now has helped us to identify the bleeding risk of patients, and also helped us to monitor the antiplatelet therapy of the patients. And with this, again, Verify Now instrument, it has helped us to reduce complications during and post intervention, and also has helped us the inhibition of high platelet monitoring. The second platform which Varfan has is hemostasis, wherein we have the coagulation products in laboratory settings. We have the systems which is called ACL top. We have a complete comprehensive menu of reagents, and all these instruments and assays are integrally connected with our latest database system, which helps us for quick and effective monitoring. So this is the ACL Top Family 50 series, which is a fully automated coagulation device. It helps us for pre-analytical modules, and in this pre-analytical module, it helps us for checking the interference of HIL, checking the interference of clots. So this is there. We have this in 3 models: ACL Top 350, ACL Top 550, and ACL Top 750, and also the latest one, ACL Top 750 LS. The best thing is that we are able to do a fast turnaround to the tune of 360 PT test in an hour. So we are continuing the advancement, and with the feedback of you clinicians, and we are evolving in this segment also. With these coagulation devices, we are able to do the general screening. We are able to do anticoagulant testing. We are able to do even tests like von Willebrand disease test and also DIC. And we have a complete comprehensive menu of hemosyl assays which are used in these coagulation devices. As far as coagulation factors are concerned, we are able to measure intrinsic and extrinsic factors, and also factor 13 antigen we are able to measure with these coagulation devices, and also von Willebrand disease as I said before. We also have a newer ACL Epistar. The key feature for this is that we are able to measure the ADAM TS13 activity, which again helps us to in the diagnosis and monitoring of TTP disease. So Werfen is now merged with the erstwhile Immucor company, and we have solutions for transfusion and transplants also. So the basic feature of transfusion is that the products which we have, they help us to maintain the compatibility between a donor and recipient. And in the transplant space, we are able to, the basic feature which our products have, they help us to maintain the histocompatibility. I'll briefly touch upon these in the next slides. And this is the rich history of our transplant and transfusion division. We started 40 years before, but in 1998, we started with a semi-automatic DIAS model. And then we evolved over the years. Now we have advanced system like Luminex in the transplant segment. In the transfusion segment, we have Eco, New Iris, and this evolution is continuing. And we are very proud to tell that our solutions are present in some of the topmost hospitals of the globe, like Mayo Clinic, Cleveland Clinic, Johns Hopkins, to name a few. Our endeavor is to work closely with all clinicians and improve and increase our solutions. So these are the two solutions which we have in the transfusion space, the EcoLumina and NeoIris. EcoLumina can be used in small settings, small blood banks where the throughput is approximately 20 cycles. And with the NeoIris, we are able to do test to the tune of 225 to 230 in one throughput. And we have various types of anti-sera which helps us for to measure the forward grouping. We have different anti-seras, anti-A, anti-B, anti-AB, and anti-D. And also we have reference cells which helps us to do the reverse grouping and map the reverse grouping. Apart from that, there are other different anti-seras which again helps us for the screening and identification of antibodies. There is a newer one, Capture Ready ID, which helps us which operates on a solid phase technology for antibody screening and identification. And all these solutions again are interlinked with our internal IT solution, middleware solution, which is called ImmuLink, which helps faster, reliable, and more accurate results. So the basic aim of our transfusion solution is that the solution should be scalable and have effective data management. In this, we have the complete range of platelet portfolios and the broadest portfolio for our traditional reagents. And the last one which we have is the solutions for all the transplant. We have the latest Luminex 200 XMAP, which again is considered a gold standard. And we also have Flex Map 3D technology. We have different type of live codes which helps us for HLA antibody detection by phenotypic method. We have live codes, beads. The latest one is, you can see the latest one is live code non-HLA antibody kit, which we are recently launching and we will be promoting actively in India. This is the latest one. And as far as genotypic method is concerned, we have live codes for HLA, SSO, live code KR typing, and also MIA4 NGS, new generation sequencing. So all these products help us to give a better solution to all you clinicians in the transfusion and transplant space. And we take pride here to stand in front of you and tell you that with all the support from all of you throughout the year, we have been able to do 1.8 million testing per day. Our endeavor is to gain further support and increase this number to maybe 2 million, 2.5 million. And I'm very sure that with our solutions, we'll be able to achieve that. Thank you and have a very nice rest of the evening.
Thank you, Mr. Ajit. It was a wonderful talk. So we have a comprehensive solution for transfusion, transplantation, as well as the hemostasis profile. So the forum is open for any questions from the audience. So the non-HLA antibodies you mean is like what type of antibodies are they? Like platelets or are they neutrophils?
So I'll request my colleague. Raja, you can come here and answer.
So over the period of time, we have observed in antibody, when clinically significant antibodies are there, and we are able to pick it during our testing on Luminex platform. But still there, we have observed that the graft, is rejected, and the reason over the period of thorough investigation and found that there are non-HLA antibodies which are affecting this. So we have got recently launched this, over the period of R&D. Recently we have launched non-HLA antibody kits, which will help you to, to get identify those non-HLA antibodies, where you can further investigate and accordingly go forward for the transplant.
That means SAB negative samples will be tested for non-HLA antibodies.
Sir, possibly, sir. Algorithm is going to come very soon for that, but SAB will always have an important part to play. But still, non-HLA antibodies will be also tested along with. So different hospitals are following different algorithms, and this will be also a part of the algorithm very soon.
One last question for your IL systems. Your platelet function test can be done. Platelets are not covered in that.
So platelet, the Verify No is there for platelet function, and currently that's a separate instrument, which is helping us for platelet monitoring. The instrument name is Verify No.
Verify No. Thank you.
Thank you.
Any questions? So we'll wait.
So when there are no questions, either the audience has understood nothing or understood full. So I will be optimistic, and the second one, I will take the second one.
Thank you, Mr. Ajitpal Singh.
Next topic is enabling blood center transformation with innovative technology for improved patient care by Mr. Said Siaul Haq, National Marketing Manager, Thermo Blood and Cell Technologies. For chairing the session, I would like to invite Dr. Veena Shenoy, Additional Professor, Head of the Department Transfusion Medicine, Amrita Institute of Medical Sciences. Madam is a reviewer of Asian Journal of Transfusion Science and Global Journal of Transfusion Medicine. Madam has won several awards for scientific poster presentation and also has published numerous articles in various national and international journals. Welcome, Madam.
Good evening, all. Let me thank the Executive Committee of Transfusion Medicine Academic Society for giving me this opportunity. Today we have talk by Mr. Said Siaul Haq on enabling blood center transformation with innovative technology for improved patient care. And he is the National Marketing Manager for India and South Asia Thermo Blood and Technologies. We all know that Thermo has been the pioneer in introducing various innovative technologies in the field of transfusion medicine. And we are all pleased to listen to what Mr. Haq has to tell us. Please
Thank you, ma'am.
Proceed.
Good evening, everyone. I represent Thermo Blood and Cell Technologies. organization is continuously enabling blood centers transformation with innovative technology for improved patient care. So I will be talking a brief about it. Before proceeding, I would like to talk a little about my company, Thermo. So in India, we are registered as Thermo Pampol, and Thermo Pampol is integral part of Thermo Blood and Cell Technologies, which is a business unit of Thermo Corporation, Japan. So Thermo is a 100 years old company. We completed 100 years in 2021, and we have three business units. Thermo Medical Care Solution, which is basically pharma and diabetes care. Then we have Thermo Blood and Cell Technologies, and then third is Thermo Cardiac and Vascular Company. So all these companies under the umbrella of Thermo, we are guided by one single mission, and that mission is contributing to society through healthcare. Our core values also remain same. We are guided by 5 core values: respect, integrity, care, quality, and creativity. Our core value is what drives our culture at work and our life at home. And all of our business decisions are being taken in accordance to the core values of our organization. Well, this slide explains connection of Thermo Blood and Cell Technologies with India in general and with Kerala in particular. We have seven global manufacturing units across the world, and one of them is at Trivandrum, and the other is at Costa Rica, Colorado, Belgium, United Kingdom, Japan, and Vietnam. At Trivandrum, we have our largest blood bag factory of Asia. And not only that, we also have our R&D center at Trivandrum. Basically, Thermo Blood and Cell Technologies have 3 research and development centers. One is in the United States, other in Japan, and third in Kerala. So this company is actually your company, it's Kerala company, and I am the employee of your company. This is the photograph of our blood bag factory and our headquarters at Trivandrum. We have more than 2,000 employees working at Trivandrum for our company. So this was brief about our company and Kerala connection of Thermo Blood and Cell Technologies, or Thermo Pampol, as we are registered in India. Now if we look at the evolution of transfusion medicine, transfusion medicine evolved from glass bottle to single bag and then to componentization, and today blood centers are transforming into cellular labs. We have been an integral part of this transformation journey from the single bag to the cellular technologies that drives the transfusion medicine industry in India and globally. Under transfusion medicine, under blood and cell technology, which is transfusion medicine driven product line of Thermo, we have 4 business units. The one is Global Blood Solution, in that we have all of our medical equipment, which I'm sure most of you must be using, and the factory is right here at Trivandrum. Then all the blood bags, we have pathogen reduction technology in this, we have fully automated whole way automation system, and we have apheresis device under Global Blood Solution. The second product line is therapeutic system, which is Optia, and again I'm sure many of you must be the proud owner of Optia device, as it is one of the most favorite device of our users. Then we have cell therapy and plasma innovation. We are in process of launching cell therapy in India. Plasma actually is, as per the law of our land, we are not allowed to collect plasma commercially. So this product line we are not going to launch in near future in India. Although there are a lot of innovation happening around plasma, we are also coming up with free-dried plasma, wherein the plasma will come in pouch, you mix the water into it and make the liquid and transfuse it. So all those technologies are underway, and it will take a little time for those technologies to come down to India. Now, coming to the stem cell, this cellular technologies that we are going to launch very soon, maybe in next 2 or 3 months time, it will be installed in India. So how it happens? The cell is collected using our Optia device, And then we have an expansion system, we call it quantum plex. When the cell is expanded using this quantum device, and then harvesting is done, and after harvesting again, the further processing is done by another device, we call it Phinia fill device. And then there are a couple of more manual steps which is yet to be automated before it is transfused to the patient. So with this technology, I believe that in coming few years' time, cancer as a disease, because a lot of inventions are on the way, a lot of discoveries and researches are on the way. So I believe in next few years' time, cancer as a disease will remain as like any other disease, and we will have affordable cure of the disease very soon with the advancement of the technology. Now, these are very kind of popular product line that we have. Once you enter in a blood center, the donor chair is manufactured by us, the blood collection monitors are manufactured by us, all the blood bags, you name the SKU and that is available with us, from a single bag to filter line bags. And then sealers, when the donation is done, the sealers are there with us. And then when we further proceed to the component lab, then the weighing scale, the automatic plasma extractor, these are all the semi-automated product line that is there with us. And if you go to the fully automated product line, then the radio system. Anyway, so that's the radio system, which is a fully automated device, followed by Tma and Optia. So Tma is again a very popular device, which is used to collect essentially in India for platelets, but otherwise it can collect multi-component, it can collect erythrocytes also, and there are a couple of centers which is collecting multi-component using Tma. Then we have Optia for therapeutic device, for therapeutic procedures. And then coming to the storage, we have all type of refrigerators, deep fridges, platelet agitator, incubator, and then when it comes to processing, we have a sterile connecting device, which is essentially used for making the therapeutic dose in collecting 2 tubes into a sterile medium. And then we have pathogen reduction technology that is, okay, so that is right there. The corner, this device, we have the first installation we have done in King George Medical University in Lucknow. This device basically, it's called pathogen reduction technology. We call it Mirasol. This deactivates the pathogens. It eliminates the need of bacterial testing into platelets. And currently, King George Medical University is using it to treat the platelets before transfusing to the immunosuppressed or transplant patients. And then we have the issuing device with it. Now, coming to the automation. So automation is everywhere, and Dr. Uma Menon ma'am talked a lot about it. Automation, if you look at the warehouses, so the upper photograph shows how the warehouse used to be and how smartly it has been automated today. Similarly, car manufacturing, if you see how it was being manufactured before and how it has been automated today. If you look at the diagnostic center, how too many tubes and different equipment, different steps were there, and now it has been automated with fully automated analyzers. Similarly, the surgical interventions has been also automated. So now the question is, where are we in our component lab? Well, the journey began with the glass bottle, and then the manual systems, wherein still a lot of our blood centers are, so India is a very diverse country. Many of the centers are like still into the primitive stage. Some are into semi-automation, and there are a couple of centers which is into automation. So essentially, we have moved from the very manual lab to automated today. So what are the challenges that we are facing into blood processing, and what automation can help us with? We face the challenge of variable product quality. The standard operating procedures of different blood centers are different depending on the technology that is being used and the product line that is being used. The ergonomics, where to keep what, process control, data management, traceability, device maintenance, that is another challenging when you have multiple devices to maintain. And then operators training. So for example, if a technician changes job and if somebody else joins, then again, you have the trouble of, training him again and getting him back to business. And the total steps is like 26 steps that we need to follow to prepare component. Well, the steps might vary from lab to lab depending on the SOP and the product that you are making. So what automation does? Automation eliminates most of these steps and only 9 steps are left out. So this whole automation device is like a washing machine. In washing machine, what we do, we keep the clothes, automatic washing machine, we set the timer, and then we go and work, do some other work, and when we come back, we get the washed cloth. Similarly, this whole automation device, what it does, it's a 4-bag system. You collect the bag, keep in the device, it automatically weigh the product, it automatically centrifuges, it automatically expresses, it automatically separates the component into different bags, and it automatically seals the bag, and then the product is ready in 21 minutes time. It's a complete walk away system. The complete challenges that we discussed in the previous slide is fully automated. And today we have 4 customers, 4 hospitals who are using it in India. AIIMS Jhajjar is one, they have 4 Revios device from us. Amrita Faridabad is another one, which is, that's ma'am's counterpart who was using Revios device. And then we have AIIMS Kalyani, they are also using this Revios device. And CMC is fully on automation with the Revios device. And with this, I think going forward, Revios is the future for India transfusion medicine component lab. So essentially what it does, it helps us to increase the productivity because it increases the use ease of use is improved, flexibility, the quality is improved. So the data I cannot share because those are already being shared by our consultant at different platforms at ISTM, ISBTI, and different CMEs. And ma'am knows it because ma'am also had a CME last time when Dr. Rahul came and spoke at your center. He showed his data, how the quality is impacting using the Revios device, and the confidence and the process traceability and everything is in control. So again, this is how the innovation that automation has changed these industries, and with automation, this is how the future lab of any blood bank would look like. Thank you.
Thank you, Mr. Hug, for that elaborate and concise talk. So, any questions from the audience regarding the topic? It's very heartening to know that you are coming up with a new technology for expansion of stem cells. And when can we expect that?
Ma'am, it's in progress. Hopefully in 24 we will have a couple of installations at some of the premium centers of the country wherein we can establish the reference center, and then from there we will see the journey, how it goes beyond. Thank you.
Thank you, Mr. Said Ziaul Haq, for excellent talk, and Dr. Veena Shenoy Madam for chairing the session. I would like to invite Dr. Veena Madam for giving the memento and certificate to Mr. Said Ziaul Haq. Now I request Dr. Kala Madam for giving the memento and certificate to Veena Madam.
Let's move on to the next session. Our next topic is paradigm shift in hemostasis and next generation digital solution for patient care. For chairing the session, I would like to invite Dr. Abhishek B. Gowda, Additional Professor, Jupiter Pondicherry. Apart from MD and DNB in transfusion medicine, Sir has also done post-graduation diploma in clinical research and certificate course in cellular therapy. Sir has bagged so many awards, honors and achievements like ISTM Young Scientist Award for the year 2019, Award for the Medical Scientist Exchange Program of NAMS in the year 2020-21, Harold Gunson Fellowship from ISBT in the year 2021, and H.T. Shori Award from ISBTI in the year 2021. And he has also numerous publications in various national and international journals. We welcome you, sir.
Thank you. Thank you. So the next talk is by Mr. Yannick Querel. He's a biomedical scientist with experience in medical laboratories of basically hemostatic laboratories, and has been working with Stago for more than 15 years now. He has been a product manager, global marketing manager, development manager, and also an international application specialist. And himself is a quality assurance manager as a medical technologist. He has worked on various programs of Stago like STA Satellite Max, Start Max, Connect.1 and Start Max 2. And also he has been involved in development of toolkit for Stago's data manager. I welcome you, sir.
I'm not on mute. Okay, so hi everyone. You can hear me now? Okay, brilliant, thank you. So just yeah. So good afternoon, good evening, everyone. I hope that the sound is okay and you can hear me well. Okay, so let's start. Today we wanted to address the paradigm shift in hemostasis and next generation digital solution for patient care by Stago. But I just wanted to tell you a story first. So in the 60s, Mr. Jacques Villeray, the owner of a French pharmaceutical company, met Bernard Chambouet, who explained the need to develop coagulation tests for laboratories and offer better patient healthcare. So they decided to develop the first hemostasis reagents in 1962 and then to create Stago, the first diagnostic company, dedicated only to hemostasis and thrombosis. So as pioneers in the IVD coagulation world, Stago is acutely aware of how coagulation, surgery, and blood transfusion are intimately integrated. So I'm Yannick Querel, marketing manager for Stago for the India, UK, and Ireland business units. And I'm really excited to be with you today and honored to have been invited for this conference. So I wanted to tell you more about how Stago's innovation meets hemostasis and transfusion labs needs today, but only today, also for tomorrow. So today I'd like to start and give you a quick overview of who we are, introduce the next generation of Stago coagulation analyzer, STHEMO, but also tell you how our digital solution bring expertise, automation, and efficiency and standardization in the lab to support clinical decisions and empower staff. So Stago is an international company entirely dedicated to hemostasis and thrombosis. From coronary assays or global tests such as thrombin generation to platelet aggregation. I've seen that there was a question about that earlier. So we have a platelet aggregation system and we are launching a brand new one this year. So more of that I'll show later. But also very niche assays such as flow cytometry assays. So basically, we know our code in and out. And for bleeding patients, we understand the importance of emergency assessment of hemostasis to determine which blood products are needed to correct hemostatic defects associated with bleeding. We also understand the crucial role of labs for hemophiliac patients during surgery for confirming the diagnosis and baseline factor levels, monitoring new inhibitors, or managing antiplatelets or therapeutic anticoagulation. But I think it's time now to dive into why this stable mission and how stable solution helps laboratories around the world. So first and foremost, a stable mission is to improve personalized patient care with powerful biotechnology solutions for blood-based diagnostics. That means Stable has a vision to improve the health of as many people as possible, and by offering reliable, accurate, innovative solutions for our instrumentation and reagents, but also via our digital solution. The current range of stable solution of our reagents and reliable instruments that meet all laboratory needs, from satellite or urgent care centers to high throughput, high volume labs, but with the same high quality results and a panel of tests that meets the needs of routine and more specialized labs. But today I really wanted to give you an overview of what's coming next. So the next generation of cell coagulation that will set the new standard in the coagulation lab. So more than 250 people have been working relentlessly on this new ecosystem for the last 5 years to work on a solution that meets your needs today, but not only today, but for the next future. And a solution that is already coming, in fact, with innovation. So one thing that has guided the team throughout the whole project is the ability to deliver the right results first time. For our patients first, but also to offer a solution with minimum friction to minimize the risk of errors for better quality results. But more than that, first, let's see what were the other key drivers for this new solution. We had 4 simple objectives. We wanted quick and predictable turnaround time. We wanted a solution that was easier than ever.
Excuse me. Hello.
Yeah.
The screen which you are sharing is getting flickering. Now it's okay. Now it is staying stationary. So when you're moving to the next screen, it gets it's moving off. Can you? Now we can see your screen, but when you're talking, it goes. Now it's gone. So yeah, now it's okay. So can you be to make it stable from your side? Can you try?
I'm not sure what's going on, to be honest. I wish I could be with you today, but thanks to the magic of internet, at least we can be together, but that means also having a few technical hiccups.
Yes, I will surely get back now. We'll be sharing the screen from here, and you can just tell me next. So when you say next, we'll move the screen. We are just downloading it maybe within 1 minute.
Yeah, no worries. The one before? One before again? Again? Yeah, thank you. Okay. So let's start again. So yeah, I was just telling you that when we developed STHEMO, we really had 4 simple objectives in mind. So the first one was to be able to deliver quick and predictable turnaround time. Second one was to deliver a system that is easier to use than ever. And then also broaden scientific expertise, bring our expertise to you in your lab with one goal, one objective for all of that, was to be able to improve patient care and patient management. So I'll start with the new STGMO family instruments. Move to the next one. And we have two brand new systems that will be launched this year. You can move to the next slide. Yeah, thank you. So we have our STEMO tool, which is designed for small to medium lab activity, and the STEMO 301 for high to very high volume labs, that will probably link to the presentation from a colleague from Terumo talking about automation and STEMO 301 is also designed for automation. So they are both powered by the same software. They share around 70% of the same hardware, and also they use the same reagents and consumables. Next slide, please. So in a context of emergency assessment of hemostasis for a bleeding patient who might need blood products, it's critical to deliver quickly and accurately and at any time at least standard coagulation assays, PT and PTT fibrinogen. So we really wanted to design a solution that never stops working, never stops producing results, whatever the situation, even if you have to reload the system and reagent or a clean solution. So it's one of the reasons why that was a priority for us to have a quick and predictable turnaround time. The question now is, did we achieve that? So next slide, please. And well, according to Peter Baker from the Hemophilia Center in Oxford in the UK, STHEMO, yes, is the first and very quick to first results being delivered and produced, which is vital for urgent care centers such as your centers. Next slide, please. So STHEMO is probably one of the fastest coagulation analyzer on the market. It's the fastest, especially for mid-spinal tests. It offers continuous loading for all the reagents, and loading is quicker than ever now with our rack-based solution. And also thanks to the gigantic 126 reagent position on board on the STHEMO 301. That means that STHEMO will never stop working, and thanks to a dedicated stat position, your bleeding patient will always be prioritized first. So we can guarantee a turnaround time in that less than 10 minutes for panel tests such as PTA, PTT, fibrinogen delay. Next slide, please. So in terms of software, I guess we all want the same thing. You, me, when we want to use the software, we want something easy to use, easy to learn, and intuitive, like we could have on smartphone apps. So that's what we had in mind when we developed STHEMO, and that's what we have done. So STHEMO offers a modern and easy to use software, and also offers a virtually and error-free solution that will make your life easier and to minimize risks for patients. So we can discover on the next slide some examples. So for routine assays such as PTA, PTT, fibrinogen dimer, there's no pipetting required and no calibration needed. So there's no risk of errors, and you have a quick turnaround time for this test. We have also removed the need of server and reducers. Again, removing that less errors and better quality results. We have a wide range of reagents. Again, no errors during pipetting. We have a short conversion time, so if you need to load reagent urgently, can be done easily. We have a long on-board stability, a wide working range as well, which also means less reduction to quicker results for the patient. We have introduced QR codes on our vials and unique vial identification number, but also something very smart, which is the automatic IOQ data transfer. So everything is stored in our cloud, and it can be downloaded directly on the system, so you don't have to worry about manual transcription, and you get all the QC ranges automatically downloaded on your system. We have also improved precision, and in the next slide you will see that we've done a study in a French laboratory in France. If you can move to the next slide, where we have demonstrated the precision of the STT-1, where Cs are all more or less below 5%, and that can be really important to have really good CD and precision, especially at clinical decision level or at the level of think about design, for example. Bringing this outstanding precision is our goal ultimately for patients, really. And that means that I will move on to the next slide to really focus on what we are bringing at innovation for patient care. So if you can move to the next slide, and if you think about trauma situation, for example, where stabilization of the patient in the first hour is so critical, one typical source of increased turnaround time in most clinical laboratories is checking sample for hemolysis. Well, with several unique mechanical detection methods, our dispositive-based detection system, which is available on all our platforms, all our instruments, you have a solution that, one, not only will detect automatically HL interferences, but two, is also insensitive to HL interference because it's mechanical and not optical. But I need to say that it's a proven method, technology, not only because it is recommended as the gold standard method for problematic plasma, such as hemolyzed plasma, by CSI H21A5 guidelines, but also because we have demonstrated that in the next slide. During a study performed at the renowned Sheffield laboratory, led by Steve Kitchen, in the next slide. Yeah, thank you. So we performed a study to assess impact of true hemolysis with spare sample, and the conclusion was that based on clinical acceptance criteria determined according to the CLSI C60 guideline, no clinically significant bias due to hemolysis with stable solution. So that's really a powerful technology to deliver quickly and accurately results in all situations. You can move to the next slide. And several dispositive detection system, that means the right result the first time for closing assays. And not only now with our VPS, dispositive detection system, but also with our brand new concept of measurements and dual technology, which combine dispositive detection system and optical measurements, all in the same pivot, all at the same time, and on multiple wavelengths. This new concept is the first and exclusivity worldwide, and that will allow us to bring in more clinical decision support for you based on algorithms. And that will help you in really complex clinical decisions in the future. And on top of that, we also bring in chemiluminescence in our system. One of the benefits would be to increase again the precision or improve again the precision at clinical or clinical decision level with CVs that we can expect to be below 2 to 3%. And we are also bringing new automated assays on our. Next slide, please. And one of the other innovations we are bringing in the next slide is that we are developing several applications based on artificial intelligence. If you can move to the next slide. So then artificial intelligence and self-learning algorithm. For example, we mentioned the dual technology before, but the first example of this application is the automatic tube tire recognition thanks to an embedded camera that will automatically recognize the type of tube and adjust automatically the belting system and check volume based on the type of tube. So that will bring standardization, less risk of errors due to tubes not filled properly, and we all know how important it is for correlation to have tubes filled correctly. So we've seen now how we've used cutting-edge technology in our automated instrument. Now we'll explore a bit more on how we brought innovation as well on our digital solution in the next slide. Can you move to the next one? So we are bringing now a brand new Sego ecosystem, STE, in the next slide. So thank you. And we have different elements in our ecosystem. First, STE Connect, which is a link in between you and us. So Sego offers a remote connection to assist you in the key, and basically the goal is to maintain your turnaround time and make sure that there's no downtime, which can be critical again in urgent situations. So with STE Connect, we will also enable the automatic download of all the package sets, such as QC ranges, calibration information. You don't have to worry about that, it will be done automatically. With STE Qualaris and Expert QC, that's our independent certified EQA program and externalized QC package. That means that in real time, you can compare your performance with the network or nationally or even worldwide. STE Sentinel is our predictive maintenance future service, and STE Manager, which is more now, is our data manager developed by collaboration experts. Next slide, please. And in terms of data manager, we have a long history with that, with this type of product, and with products suited for every lab and every need. Next slide, please. And we have solutions for a single lab, for all, bringing everything, our QC package and data manager experience into your lab. But also in the next slide, we see that some labs in a networked situation can face challenges. And our STE solution will help you meet these challenges and effectively manage data across the network. Next slide, please. So first, STE Manager will bring you one single database, either if you are working in a single lab or in a network, and smart auto-validation rules and clinical decision support. You can click on the animation. Thank you. So the second element of STE Manager is our quality package, including a complete comprehensive quality solution for you and accreditation tools. We have also full traceability with all the results that can be auditable, and using state-of-the-art technology. So that means we are bringing service resilience with no single point of failure for your lab. Next slide, please. And now we see how it can help you with standardization, automation, and efficiency in lab with the next slide. And you can click on the animation element. So with our colleague from Telemore before talked about standardized SOP. So we can help you with that by automating your current SOP. Let's say you have a prolonged PT, you want to first rerun it and then make another test. We can do that for you. We have a set of rules for reflex testing, repeat testing, automatic expert evaluation, but you can also pick and choose from a published expert rule library that Segway validated. If you can click on the animation again. And all the benefits of data management that are available in different studies are all the same. We decrease turnaround time, decrease cancel time, and standardization. And I'm just focused on turnaround time today because you can save 6 minutes per test using automatic validation rules. That means it can be a critical time for some patients first to give results, but also with all of that. The next slide. When you ask our customers, for example, you have a customer in Belgium. In the next slide, thank you. Where he has implemented automatic validation rules and he said that 88% of all the patient results are automatically validated. That means saving time for the technicians. That means you can focus more on patients that deserve more attention, but also that means you have more time to implement new assays for lab and develop your service. And thank you. We also wanted to take a step further because we know hemostasis are it's complex matter and some labs need a bit of help with that. So we have developed a complex algorithm. For example, we have a factor inhibitor algorithm that has been developed with LabCorp in the US based on care recommendation. So it's a fully automatic factor inhibitor algorithm using 117 rules, just to tell you how complex it is. And they have truly seen an improvement in quality of results in that lab. They also mentioned that it can be a really significant benefit in low volume labs with staffing problems and also where specialization can be a challenge. And we are also bringing a display of algorithm for next slide, please. The other algorithm we worked on was one developed by Dr. Sukesh in India, around lupus anticoagulant that has been published in the Journal of Blood in November 2022. And its conclusion is clearly gives a better clarity in classification of patients into true positive and true negative, and a better detection of interference due to paper. Also seen a 60% reduction in cancel time while maintaining the same cost. The study also concluded on the importance of mixing studies in avoiding false negative. Next slide, please. And we have seen also that we wanted to take a step further. So the next step for us will be to rework the existing lupus anticoagulant algorithm. So we have another one published in ISLH in 2018. And we are bringing telemedicine on STIMO and the powerful rules in STIMO manager to build and develop and enhance this algorithm and bring cadolipine and beta-2p1p to have a complete diagnosis of antiphospholipid syndrome. So we are working with a group of experts around vascular platform, Synapse, the strong generation expert, and Catherine de Vries from Kent University Hospital, who is an expert on lupus and working in ISTH subcommittee. The next thing where STD management really help is know that labs face a burden for them to meet the demanding ISO 151 requirements. So Stable Accreditation Toolkit that is embedded will help you with that. So we have a set of tools where basically you can run your test on and the results are automatically uploaded into your data manager. And Stable Accreditation Toolkit will automatically produce reports that are compliant and that are including all the statistics you need. So we have a few examples in the animation. So we have tools for repeatability. If you can click once. So repeatability, the next one is correlation, comparing between instruments, but also for batch acceptance testing, comparing the old batch and the new batch. You can click once. Yeah. We have another one, the next one is reproducibility and QC batch acceptance testing. We also have the next one, contamination and linearity. And the final one, which is a complex topic, it's measurement of uncertainty. It can be difficult to calculate if you want to click just once. But our manager will do that automatically for you in real time. So it's not a burden anymore to do that. But don't take my word for granted. In the next slide, we've asked them cameras from the Royal Sussex County Hospital in Brighton, UK, which is sort of our accreditation toolkit. And she said that far more than just innovative software, steroid accreditation toolkit just simplifies the complex. And again, from the next slide, from Ellen Lewis, Hemisphere Manager, Basingstoke in the UK, said that it simply does the hard work for you. It's just to tell you how powerful these tools are. So in the next slide, we've identified really that we think there's 4 key elements to enhance laboratory stability. You need robust, accurate, easy to use analytical platform with reliable turnaround time, an IT solution that really brings standardization, automation, and expertise in your lab. You need easy way also ensuring accreditation compliance and also making sure performing monitoring quality in your lab. And that's where STE-Mode and STE-Meet in the next slide will truly believe that will help you enhance your lab and really believe that it will help coagulate blood transfusion laboratories to quickly and easily deliver the right result the first time when it matters, and also provide clinical decision support in more complex situations to ensure an improved patient journey and clinical outcome. I just wanted to finish on the next slide around our educational program. We truly believe that the more staff in the lab understand coagulation, the better it will be for the patient. So we have set up really education program from our webinar to our podcast as a podcast, but also iPad and smartphone apps such as Tmascon, where it's more for clinicians, so you can put your patient detail and it automatically calculates, for example, the 40 spots. So thank you all for your attention today, despite the few technical problems. I hope that you enjoyed it. I did further and enjoyed to be with you. And I guess it's time for questions if you have any.
Thank you. Thank you, Mr. Yannick. First of all, I would like to apologize from the side that for all the technical hardships that you had to face, and also few audience on the, yeah, thank you. That was a nice presentation.
Thank you.
I would see if anybody from the floor wants to have a question with you.
I have heard that an equipment called Quantra is coming up from Stego. Quantra, it's a point of care testing for coagulation. So
Yes.
can you just like any differences between or clinical applications, any studies that have come out from that part of the world?
So yeah, the control or display testing point of care device that can be really helpful for its here during hemorrhage episodes. So Contra has been launched in different markets, but I'm not part of the team launching it. worldwide, and I'm not sure exactly when or if it will be available for the Indian market.
Okay, thank you.
Thank you. I don't see any more questions. Thank you. Thank you for the excellent presentation and also bearing us with all the this one.
No, again, thank you for your attention. I wish you the best for the evening, the weekend, and hopefully we'll meet soon this year during one of my visits.
Thank you and good night.
Thank you. Bye.
Thank you for the wonderful session, Mr. Yannick Kurel, and thank you, Dr. Abhishek B. Gowda, for chairing the session. Now, I would like to once again invite Dr. Kalam Madam to present a memento to Dr. Abhishek sir.
Now, I would like to invite Tmas Secretary, the most diligent and exuberant member of Tmas, Dr. Abubakar Muhammad Rafi, Associate Professor in the Department of Transfusion Medicine, Jubilee Mission Medical College, for delivering the vote of thanks.
So I would like to take this opportunity to thank each person who has come over here. Like it's my responsibility to say as a secretary and organizing secretary of this event. So first of all, I would like to thank all the faculties who have taken time to come over here from Rema ma'am, both Rema madams, Yannick Korel who had joined us virtually, Ajitpal Singh from he has joined, then Dr. Charmaraj sir from Ortho OCD and Dr. Saeed Zayadul Haq from Termo. And our dear chairpersons, our own members, like Sushila madam, Maya madam, Veena madam, Nitin sir, Mohan sir, and our own Abhishek sir. Thank you for taking time off work and coming over here for chairing our important session. And our it's rather than a CME, we were thinking about bringing it all families together so that we get to know each other. And from the beginning, like 2 or 3 months back only, we started planning on how to have this event. So we, from among ourselves, including postgraduates, we made teams and we made like CME who will coordinate. And so all this thing which you are seeing today are because of the all my faculty, like my PGs, my friends, all who have come together to make this function. That is, we have initially a CME in which scientific session is there, and after 7 p.m. our own family will be coming together as a family meet in which our members and our kids will be performing. And tomorrow morning we have a PG oriented session, like oral presentations are there, and next there's a quiz, and at the end we have an annual general body meeting in which we have the transfer of executive committees to the new vibrant committee. So these are our coordinators who took this session forward, and without all our delegates there is no point. Like any CME or any program becomes a success if we have people to join only. So we have, like, this is being mainly a residential event, we have around 40 rooms, 40 different families attending this program, and around 10 to 15 other delegates only for the CME alone. And last but not the least, our sponsors, they have taken their time and effort to bring in their own faculty, like to showcase what all they have, rather than most of the other CMEs. What we see is that we or doctors would be sharing their experience using these technologies. So we thought, why not they itself come and share what they have in hand or what is in store for them. So I would like to thank Kudal Ortho, Roche, and Hemlibra for the hemophilia related talk. Werfen, as of now, we know that Werfen has 2 parts which is of use for the transfusion, that is both of Immucore and also for the hemostasis part. Then Termo, which is a big name in blood banking, they have their own factory in Trivandrum, and Stago, whose talk we just now heard. So thank you all the sponsors for the CME. Thank you. And our audiovisual team, Reni, Shaudi and AM, and also from the Crown Plaza team. Thank you each and everyone. Bye-bye. So after this, we'll be having the family meet session, which will be starting at 7 p.m. You can all go and just refresh and come back. And in the meantime, we are also planning a photo shoot using our t-shirts, which was given. Whoever has got the t-shirt and who has can get inside, we can take it for 6:40 or 6:50 p.m. We can have a photo shoot. Whoever is available will take. By 7 p.m. we plan to start the family meet here itself. Because people they want to change again t-shirt to the normal dressing. So we will disperse for now. Just see the locality. This room will be kept aside for they have